I think I've got the wrong color shirt today.
It's wonderful. Hi, how are you?
Good.
Hi. You can see us.
Good, okay. I'm Jessie Herbert. I'm with the University of Montana, and we are Montana in the Classroom. Thanks for being here for Brain Awareness Week. And this is Sarj Patel. He's a...
sup.
There's a body there.
There's somebody.
Come join us.
Sarj is with the College of Health Professions and Biomedical Sciences and does some amazing research that he's going to share about today. So thanks for being here.
Thank you, Jessie.
Perfect. All right.
Yeah.
Should we get started then?
Let's go ahead. And if Bozeman comes, we can just quick catch them up. So.
All right. Well, thank you for all being here today. As Jessie just said, my name's I'm Dr. Sarj Patel from the University of Montana, and I'm a researcher in the Department of Biomedical and Pharmaceutical Sciences. And that whole name thing is a mouthful, so I usually go by Sarj. So if you have any questions or if you have any queries, stick your hands up. I can pause. Feel free to pipe in if there's something I'm saying that doesn't really make sense. That can happen. I realize I have a little bit of an accent too, so if that causes problems, please let me know. I do use some funny words. But to get started, so, as part of Brain Awareness Week, they asked me to speak because regardless of the fact that I'm a professor of molecular pharmacology, I'm also considered a neuroscientist. And before we get rolling, and I'm sure you guys all have a pretty good idea of what neuroscientists are supposed to be, but, my path to becoming a neuroscientist wasn't as straight as you would think. It's not just being in science. I actually started out as a biochemist and a pharmacologist. When I was in high school, I loved biology and chemistry. Used to get me into all kinds of trouble, and I was able to go to university and study pharmacology in London. And I did my master's degree actually in neuroscience there, and I ended up in Montana to do my PhD in pharmaceutical sciences here at the University of Montana. And you can see behind me, lots of different professions can be classified as neuroscientists depending on what they're working on, what they're actually doing. And my background in pharmacology, my background in chemistry, biology, has really sort of allowed me to work into everything that makes everything from the neck up work and how the brain functions. And so some of the stuff I'm going to talk to you a little bit about today is the work that goes on in my lab that's related to traumatic brain injury, and more specifically concussion. Concussion is a type of traumatic brain injury. It's just people have different ideas of exactly the severity, what causes a concussion, but what causes, what is a traumatic brain injury. Anything that impacts your brain can be considered a traumatic brain injury, regardless of how slight that injury is. Before I get really rolling, has anybody in the audience, just show of hands, had a concussion or traumatic brain injury? Do you guys play sports?
Yes.
Yeah.
Yeah. What sports do you play? Just shout out real quick.
Basketball, softball,
Basketball.
volleyball.
All right. Those things will do it.
Shouldn't can you unmute your microphone and chime in?
Who, me?
Yeah, I want to hear you guys too. Can you hear me?
Shouldn't can you hear us? All right. Out of curiosity. Oh,
Okay.
there we go.
All right,
yeah.
good.
Is it? Okay. Hello.
All right.
There they are.
There
So,
you go. Now we can hear you.
do you want to tell what sports was the question? So Chinook, do you want to chime in on that one?
Tennis, basketball.
Okay. Have any of you guys had a concussion?
Yeah, a couple of them have.
A couple of them have? Okay. How long were you guys out? Were you unable to play for a little while?
Few days.
Few days, just a few days. All right. So that's really interesting because some of the stuff I'm going to talk to you about today is trying to get a better understanding of exactly how long your brain is really injured and what we can use to look at those injuries. Oh, looks like we have Bozeman coming in too. All right. I'll keep. Can you hear me, Bozeman? Yep. Okay. I think they're on mute too. All right. So, traumatic brain injury. There's more coming. All right. Traumatic brain injury and concussions is a really, really big problem, and it's becoming recognized as a pretty large global health issue. This slide here just shows if we look at the world and the population, there's about 200 recorded TBIs for every 100,000 people on the planet every year. And if you think about being 6 billion people on the planet, roughly, that's a lot of concussions, a lot of TBIs. In the US alone, they estimate, the CDC estimates there's about 1.7 million people a year within the United States that suffer these injuries. And the cost of treating and all the health care and all the other social issues related to these injuries, they believe costs about $77 billion a year. One thing I'll say, this number is probably a very low estimate of the true number of injuries occurring here in the United States, because this is based on hospital visits. Just again, show of hands, the folks that already said they'd had concussions, how many of you guys actually went to the hospital? Just one? Two? Okay. So you can see just within the class, all the folks that said they had concussions, but which ones actually made it to a hospital based on potentially their severity, there's a big disparity there. And so we have to really watch these numbers. And then the last line there shows how the state of Montana fares in the nation with relation to these injuries. And it turns out we're actually second in the nation when you look at the number of TBIs that happen for the population per capita in the state of Montana. So about just over 1 million people, and there's about 130,000 people, about 13% of the population of the state that are currently living with issues related to traumatic brain injury. Again, this number is very low compared to what we actually think the numbers are because of reporting. And so, very quickly, obviously, what is a concussion? What is a TBI? And I mean, it seems obvious, but it's basically the transference of force to your brain. Okay. TBIs or concussions are acceleration, deceleration injuries. Okay. You are moving. The human subject is moving at speed and then comes to a rapid stop. Okay. And if you think of the skull and the brain and the fluid as an egg, you've got an yolk, which is like a ball of fat, which is essentially what the brain is, suspended in a liquid sac of water, the egg white, and then encased in the skull. And you can see when you rapidly even shake an egg or hit your head, there are coup forces and counter coup forces that cause the brain to basically rattle and bash up against the skull inside your head. And obviously that causes an acute injury, as I've written across the bottom there, but some of these injuries can have some very long-term effects. And again, I'll get to some of that stuff as we go on. So saying that, and I'm just going to let you guys chime in real quick, what do you think are the most common causes for concussions and TBIs? I know there's some very obvious ones. Just shout out. Say that again, I couldn't. Sports, obviously, yeah.
Kicking your leg. Car accidents.
Car accidents, good. Car accidents as well.
What?
I mean, those are
Oh, falling.
absolute number one, falls. So if we look,
Nice.
more comments, the most common causes for these kinds of injuries are falls. It's usually somebody standing up, going from this height down to the ground. It's amazing the number. I mean, obviously children and elderly have those issues, but it affects a lot of people. Vehicular accidents are another reason, though those are actually coming down. Any guesses why?
Seatbelts.
Seatbelts, exactly. That's exactly right. And then collisions, not in vehicular accidents. Obviously, sports comes into there. And like I said, there's about 170,000 recorded sports-related concussions a year. And then, unfortunately, assault is one of the issues.
All right, hold on. I'm gonna do it for them.
Okay.
Just keep going.
All right. So obviously, these kinds of injuries have an impact on probably the most important organ in your body. Obviously, I'm biased, and so when we look at the brain and we look at what is going on. I mean, you have to look at the organ that this injury is impacting, and this is probably an understatement, but the brain is extremely complex. Okay. The most common analogy made about a brain is it's potentially your computer, right? It's like a computer chip. And if you look in the world of computers, the most powerful computer chip at the time I made this slide was called the Xenon Phi. And, computer chip's power comes from the number of transistors, which are like the electronic gates in a computer chip. And the most powerful chip at the time was 7.1 billion transistors. It's a pretty powerful chip. If you compare that, I'm going to scoot across this way. If you compare that to the human brain, the unit in a human brain that's similar to a transistor is called the synapse, and it's basically the connections formed between two independent or individual neurons in your brain. And if you look at, so this is just one region of the brain. This is called the hippocampus. And these are individual neuron cell bodies that are lit up because we've introduced fluorescent proteins that glow different colors depending on which proteins are being made. If you look at all these hair-like structures, well, I'm getting out of focus, they're all the connections. And if you look in the human brain, the number of those connections is about 100 to 500 trillion synapses. So you can see several orders of magnitude much, much higher than a computer. And so, obviously something as intricate as this, a blunt force injury like traumatic brain injury, we would think would have some pretty gross effects. And if we look at imaging techniques that can look at how connections between the neurons are changing, we can use techniques like this is diffusion tensor imaging. So has anybody had a CT scan or a MRI scan before? Just show of hands. Okay, so you've sat in the machine, big magnets around you, and it's taking pictures of your brain. Well, this is a very similar technique, and it's actually using the water in your brain to define where all the connections are. Okay, and so if you look at this brain right over here, this is a 45-year-old male who has no history of neurodegenerative disease or brain injury, and you can see how the connections from different parts of the brain are formed and how full they are, the number of connections from the front to the back. The colors indicate also the number of fibers running in those tracks. So you can see across the brain, across the two hemispheres, front to back. I mean, there's some pretty strong connectivity from the different sides of the brain. If we look at this gentleman, this is also another 45-year-old male who suffered multiple traumatic brain injuries. Okay, and you can see following the TBI, what's the biggest difference?
Bozeman, what do you think?
Less connection.
Yeah, it's as simple as that. There's a big drop in the number of the connectivity of the brain in this instance. Okay, and so we can see gross changes like this on imaging, but what we are really trying to get down to is what are the molecular mechanisms that are responsible for these kinds of changes? Are these changes permanent? Is there a way that we can get this brain to look like this brain again? Or is there anything we can do to intervene to make that happen? And so as we do the work here at the University of Montana, obviously we need to be able to look at traumatic brain injury in model systems. And for us, if you look, when you get an injury like this, you can see behavioral outcomes in human subjects. We need to be able to figure out what's going on at the level of those individual neurons and the circuitry within the brain. And so basically what we use there, obviously, is an animal model. And this video is just quickly highlighting the regions of the brain that we're very interested in, areas like the hippocampus, this dentate gyrus, because those are areas that are really involved in learning and memory. And the activity of the neurons in this particular region of the brain are extremely susceptible to traumatic brain injury. And so this image is just sort of, this video is just sort of highlighting the regions of the brain that we're very much interested in studying. And you can see it's continuing to zoom in. So we've gone from the structure, now we're starting to look at the individual neurons here. And the region of neurons that we're interested in are called granule neurons, a specific type of neuron in your hippocampus. And this video is basically just showing them, obviously how they fire and how the connections all work. And this basically highlights the single unit that we're very much interested in. It's a granular neuron. These hairy spine parts here, this is the dendrites, this is the axon, this is the part of the neuron that collects information from other neurons, so signals from other neurons, and then it passes those messages down the axon to its neighboring neurons. So how do we do the TBI? All right, so we use this instrument called the lateral fluid percussion instrument. Okay, and it's basically a way for us to generate a shock wave that we can pass to the brain of a rat or a mouse, so we can very accurately replicate injuries. One of the things in science, when you look at TBIs in humans, obviously they're very, very different depending on the force of injury and where the injury occurs, what side of the brain, what side of the head, the amount of force. For us to be able to figure out exactly what's going on and commonalities, things that could be changing, we need to have a reproducible model for looking at TBI. And so we use this instrument, and basically what it is a saline-filled chamber. This is like a big chamber of water, and there's a pressure transducer here that's connected to the skull of the rat. And then basically this is a weight that we can draw back, we can release, and it sends a pulse of energy through this fluid into the skull of the rat. And we can very accurately measure the amount of force and the time over which that force is being applied to get a very reproducible injury. And so this is work that's actually happening in our labs here at the University of Montana. Obviously, I don't do this by myself. I have a really good crew. I apologize for the speech bubbles, but I couldn't find the undoctored version of this. But this basically shows my technician, Diane, and she's working with a high school student from Missoula County Public Schools here in town. And what she's doing right here is generating slices of the brains from the animals that we're studying our TBIs in. And what she's doing here is, this is a paraffin-embedded brain, and that little slice right there that she cut is 10 millionths of a meter thick. So we can start looking at changes in that brain and looking at things like the proteins and the RNAs in there for the studies that we're doing. And so these are some of those slices. So some of the slices that were being cut We're very interested in this protein called XET. It doesn't really matter what the protein is. It's just an example we're using in this one. I'm interested in this protein because it's very important in understanding how the brain responds to injury. Okay, and obviously in a traumatic brain injury, this could be an important protein. If we look at this, I keep doing this, if we look at this slice here, this is a control rat. So this is a rat that has never received an injury. And this is a coronal slice, sort of midway through the brain. So a coronal slice, so if you go from nose to back, sort of about halfway through the brain, okay? And you can see on this slice is the cortex, the important areas for memory, hippocampus, and there's two of those on both sides, and then this area right here is the striatum. Rats don't have temporal lobes, so we don't see any of those. And what we're looking at here is the protein I'm interested in lit up with a fluorescent tag, okay? So I've got an antibody that has this red fluorescent tag on it, and so wherever that protein is in the brain, when we put the antibody on there, we can actually see where that protein is. And so this is a control animal. This isn't in green, this is the same slice, but I've tagged it for another protein and I've labeled it green. So I can look at the two proteins in the same slice at the same time. And this one is known to change during injury. So I've got a control built in so I can see how my injury is affecting the brain. So this is the control. This is a slice from the same area of the brain from an animal that received a traumatic brain injury 48 hours after the injury. Okay, and if you look at the two, obviously there is an incredible difference in the amount of this protein being expressed throughout the brain in this animal. And that was really intriguing to us because I've studied this protein for a very long time, and in most cases, if you look in things like the liver, the kidneys, the lungs, if those cells are injured, we normally see this protein go up, okay, in response to injury. That's what it's supposed to do. But for some reason in the brain, we don't see that, and we're not entirely sure why at this point. We wanted to know, how long does this change stick around? Is it permanent? Is there ability for the protein to come back? And when we look 48 hours out, what we see is that there's some recovery of the protein. Okay, it's starting to come back. It doesn't come back in the same manner as this one over here. You can see that the localization of the protein is different. So what this is telling us, quite simply, is that there are some pretty major changes happening in the brain, even though the brain itself looks normal. Okay, there are some molecular changes. So if you were looking at this brain and that brain on a scan, just like a CT scan, you probably wouldn't notice too many differences. But this is telling us that there are some pretty huge molecular changes. And if we look at this brain and then compare it to this brain, you can see this protein shows where cells have rapidly divided and are responding to injury. And if you look right here, this region right here, in our instrument, the shock wave comes straight down. It causes the brain to twist. And this region is what impacted the skull. And you can see the scarring. And that's reproducible in our model. And every time we do this, we look for this, so we know we're getting the same injury. This is sort of like looking from a plane down on this slice. If we zoom in and look at that specific region, the hippocampus, you can see these are those individual neurons I was just telling you about, those granule neurons. And this is the control slice here, and you can see where my protein is distributed. Okay, you can see it's around the cell bodies. Each one of these blue dots represents a neuron, a cell. And then these are the fibers that come off, and this is where my protein is. If you look in the injured animal, you notice that protein staining is gone, and you can see where the localization has changed and these fibers have become much, much narrower. So this was fairly new. No one has ever shown sort of this kind of change before in a protein in TBI. And so that got us thinking, well, what could be, as scientists, we're trying to figure out what could be responsible for these kinds of changes, knowing the mechanisms that are involved in expressing this protein. One of the things sorry, go.
I was going to say, I want to pause before we go on too much further. And I just want to see if they have any questions at this
Sorry,
point.
I keep going on. You guys got to interrupt.
It's fantastic. But yeah, why don't we just go through each of the schools, and I just want to see if they have any questions about what we've talked about so far. So, do we want to start with Chinook?
Nope. We're good. We don't have any questions.
Okay, Chinook's good. How about Cutbank? How about you?
What makes a person susceptible to having a concussion? Right?
Susceptible?
Like, do different people more susceptible to receiving a concussion?
Absolutely. That's one of the things our research and other researchers around the world are starting to find in our work. What we're noticing, obviously concussion became a big thing primarily because of sports, okay? And as you guys have already attested to, you get a lot of individuals that can suffer concussions, but not all of those people go on to have long-term effects of concussion, based on the number of concussions they have. Maybe there's a threshold. If you had 8 concussions in your playing career, and I'm making that number up as an example, up to that point you may be able to recover fully and never have any long-term effects. But some individuals can get 3 concussions, and then the way their brain reacts, we see some really long-term detrimental effects of those injuries. And that's some of the work that I'll describe here in a little bit. We're trying to get an understanding of what makes, person A susceptible, but person B resistant. And that's some of the research that we're trying to do to try and figure that out. Good question.
Yeah.
Thanks.
Bozeman, do you have any questions?
Don't
What are
be
you
shy.
going to do with this information?
Say that again?
What are you going to do with all this information?
I'm gonna tell you here in the next minute. Not to give too much away, and I've got some questions for you later. We're trying to use this work to get a much better understanding of, how long these injuries are persisting and exactly what's changing. And, we're looking at one very small part of the whole injury. We're concentrating on a set of genes that we're very much interested, but we have researchers all over the world that are collaborating with us that see some other changes, and we're trying to figure out how all of those things work together to either help the brain repair or hinder the brain from repairing. And so we know where we need to target our efforts to try and know which genes and which proteins need to change to improve the outcome for TBI and concussion patients. All right, so are we good? Any more questions?
I was wondering if you have multiple concussions, will the protein in your brain, will it ever come back how
Great,
you were before?
great question, and that's exactly what we're doing right now. So we've developed models to look at multiple injuries. Very few people, especially if you're looking in the arena of sports or on the battlefield for the military, they very seldom receive just a single injury. Okay, and what we're already starting to see in some of the work that we're doing here at the university and in collaboration with our colleagues around the world is it's not just that there's a difference between lots of injuries and a single injury, and the outcomes do change. And what we're trying to get a handle on, obviously everyone wants a magic number. What's the minimum number before things become irreversible? We don't know. It's going to, again, as different as everyone is, it's going to be difficult to figure that out without doing some huge clinical studies. But as we move towards that, some of the molecular changes that we're seeing are giving us really good insight that there may be a threshold, or there may be, so much damage you can take before you reach a tipping point. And we're trying to find those mechanisms. We actually think we have a handle on one of those, and that's going to you give me a nice sort of segue into the next section. So, these changes are occurring, and we've not seen these changes occur in these kinds of injuries before. So we had to try and figure out what potential mechanisms. And I'm going to make a bit of a leap here, but I'm going to try and explain to you why we think these particular molecules I'm going to talk about next might be very much involved in the way TBI affects a human brain. Okay, and so those molecules are microRNAs. Okay, and so I'm going to get out of the way here. So what microRNAs do is basically they're very powerful regulators of the way messenger RNA gets turned into protein. So as you all know, all your genes code for all of your proteins on the DNA. When you want to make one of those proteins, it first has to be transcribed off to make messenger RNA that codes for that protein, and then you get translation that makes the brand. So this scheme I'm sure you're all aware of. What we're finding now, microRNAs are a group of molecules that come from the DNA. They're just like genes. But what they actually do is they interfere with this process of turning RNA, which is the signal for proteins, into the actual proteins. Okay? And the reason, obviously, it seems pretty obvious. It took a little bit of work to get here. We were seeing the loss of a protein, right? I just showed you those slides right side by side in 24 hours, 48 hours. We saw a big change in the amount of red on our brain slices. And we were starting to wonder if these molecules might be responsible for those changes. So it was a new way to look at some of these injuries in TBI. Okay, and I'm not going to really go into this, but what we found is when we looked at some of those very same brain slices to see if there was changes in the amount of microRNAs compared from uninjured animals to injured animals, we actually found a whole host of microRNAs that were going up. Okay, and so one of the things that we then asked is, do microRNA have a role in TBI? Okay, and so this became a very important question for our lab, and sort of took our project into a new direction. And I'm just going to outline some of the work that was done here. And this work was actually done by a high school student in my lab. Her name was Caroline McCormick, and she was taking the Advanced Problems in Science course here that's run here at the high schools in Missoula. And she basically came into my lab, usually missed sixth period at school, to come work in the lab in the afternoon to answer this question. Do any of these microRNAs that we found change in our rats? Can they actually change the expression of the protein that we were very interested in? Okay, and so she would come in, and she was working at the University of Montana in the Health Sciences Department, and I work also in the Center for Structural and Functional Neuroscience, is where she did most of this work. And basically her experiment was, we know the protein that we're interested in, okay, and this is a model, a molecular model of exactly what my protein looks like, okay, and this is in the membranes of all those cells, okay, and what she did was, she wanted to look to see if those microRNAs could affect how much of this protein is actually being expressed. And to start off, obviously we didn't start in a whole brain. That's, it's a pretty difficult experiment to do. So what we had Caroline do, and what she did, was she used a cell line. Okay, so she would grow human brain cells in tissue culture, and so this is actually one of her flasks. So it's filled with media, and on the base of this flask are glial cells from a human brain. Okay, and what she did then was, obviously we were really interested in these microRNAs, and these microRNAs are small pieces of nucleic acid, So what we had her do next was, this is what the cells look like at the bottom of the dish. I know it's probably difficult to see on your screens. Okay, what we had her do was she introduced the microRNAs into the cells. So she was basically, what she was trying to do was, we make the hypothesis that these microRNAs are going up, and they're going up, they're affecting the cells, and those cells are making less protein. So she tried to do that in a dish, basically, to see if our idea was correct. Okay, and so I know it says D up here, but this panel just shows cells that weren't transfected with our microRNAs. And if you look here, what we actually did, or what she did, we used a molecule for our microRNAs that had a green tag on it, another fluorescent tag. And you see all these green dots over the cells. That means that the microRNAs that we're interested in, or she was interested in, were getting into the cells. Okay, so she did these experiments. So what we did then was, after these cells sat for a little while, and we could do 24 hours, 48 hours, 72 hours, as long as we wanted to, as long as the cells remained healthy. What we had her do then was use this machine up here, affectionately known as WES, to look at the proteins, or the specific proteins that we were interested in, just to see if adding the microRNAs could make those changes. And she had some, pardon this, smoking hot data, actually. It was really, really good. So this, we were very excited, because she's the first person in the world to show this. She was able to show, so this is a Western blot. And I know it just looks like a bunch of dark blots on a white background. But what this method allows us to do is, she can take these cells, okay, and make a lysate. So she basically takes them off the dish, and she can extract the proteins out of those cells, okay? And what we can do is we can load them on this machine, and what that machine does is separate all the different proteins by their weight, okay? And then we can use a very specific antibody that only recognizes the protein that we're interested in to see how it changes, okay? And so what this blot is showing here, this line right here, is our specific protein, okay? And under different conditions, and I'm not going to bore you with all the different conditions, you have to do a lot of controls to make sure what you're seeing is real. It's not due to because I'm using a transfection reagent, or it's not because I used this piece of DNA, or you should use all kinds of negative controls, like pieces of DNA we wouldn't expect to work, or RNA we wouldn't expect to work. Long story short, when you take this data and you transform it, what she was able to show, okay, was these are cells where we just put the chemical on that gets the microRNAs into the cells, just to make sure that that doesn't have an effect, and it doesn't. We do a negative control, so this is basically like a nonsense piece of RNA, a microRNA that doesn't really affect anything, and you can see it doesn't really change the cells, the expression of the protein in the cells. Okay, so if you look at these two bars, that's like our 100%. That's how much protein should be there if we don't do anything to the cells without an injury. The first one she tried didn't change anything, okay? But some of the other microRNAs that we found from our initial study, she showed that you could see a big drop in the amount of protein, okay? So this is the first data that we've generated in our lab that actually shows that those molecules that are going up after a traumatic brain injury can really affect the expression of that protein. And it sort of ties back in to what we were seeing in those rat slices when we were looking at the expression of the protein. This is much more quantitative, though, because we can look and get a real idea of exactly how much protein is changing. Does that make sense? Did that make sense to everyone? Does anyone have any questions about any of that?
Let's start, like, we'll go backwards this time, so we'll start with Bozeman this time.
Where do the microRNAs come from?
Good question. So the microRNAs actually come from the genome. So the same place that all your proteins are coded for. Okay, so microRNAs work like genes, they just don't make proteins. Okay, and what we're finding out, and as people far smarter than me have shown, is that this is another level of regulation in the human body, not just in the brain, but for all of our organs. And it's a pretty relative, 10 years seems like a long time. I know for you guys, it probably seems like forever. But 10 years in research isn't that long. And, it's taken about that much time to start to understand exactly what the role of these microRNAs in any system, not just in health, but also in injury. And, people are starting to look at these molecules in all kinds of injury models. Obviously, cancer is a big one. But for us, traumatic brain injury, this is sort of some of the first data to show that changes in microRNAs can directly affect proteins that are changing in TBI.
Good. Why don't we move to Cutbank? Cutbank, do you have any questions for Sarj?
Okay, for contact sports.
I'm sorry, I didn't hear what you said.
Are you an advocate for contact sports?
Good question. I get that one a lot. Okay, so I'm going to be honest here, and I'm going to give you a disclaimer. Some of the work I'm doing right now, my lab got funding from the NFL and GE as part of an initiative they had to look at concussive injuries. That being said, both my kids play sports. Okay, I have a 13-year-old and an 8-year-old. My daughter plays soccer. She lives for soccer. Doesn't matter what I tell her, if she can head the ball, she's gonna go head the ball, even though the federation's saying don't do that if you're under 11. I enjoy sports as much as anyone. I don't think getting rid of contact sports is a good idea. Okay. What I think we need to do a better job of is understand what these injuries actually do. And secondly, more importantly, when is it safe to return to those activities? Okay. Just this little bit of data I'm showing you, that one slide where I showed you at 48 days, this protein is trying to come back, but it's still not all the way back. Okay, so the brain is still not back to a normal state that it was pre-injury. Okay, if you look at that in a rat at 48 days, that's like a 6-month time span in a human. And I'm making some assumptions there, but when you look at the age of a rat, the lifespan of a rat, and you compare that to a human, it's some pretty long time points. So I guess what I would say is while I'm a proponent for contact sports, we need to do a much, much better job of knowing when it's actually safe for participants to go back. So if a running back gets injured, the NFL might not like me telling them, hey, your running back can't play for the next 16 weeks, which is an entire season. Right? That's going to have an impact on the sport. How they deal with that, we're not sure. Okay? We're just trying to give them the best information we can so they can make sure that when people actually do return to activity, and it's not just sports, people in the military, workplace, in Montana, ranching, farming, really high incidence of traumatic brain injury, TBI injuries. And we need to make sure that for all citizens, we know when it's safe for them to go back. And so I would say, I'm okay with contact sports. We just need to know when it's safe for those individuals to go back and do that. And some of you guys have got great questions. You keep preempting everything I'm going to talk about. But one of the things that we're trying to do is get a much better handle on when is it safe. Can we find ways to look at that?
Great. Let's ask Shanook.
Maybe one more.
Oh yeah, go ahead.
Okay, so have you or either of your children ever had a concussion, or what sparked your interest in studying this? What interests you in wanting to know, find out information about this?
Good question. As I was saying earlier, I had a love of science from when I was in high school, and I've worked in lots of different fields in science. Okay, I didn't actually start out doing anything with the brain at all when I first started, but when I did my master's degree at King's College in London, I got very interested in brain tumors. Okay, and so I started my interest in looking at how the brain functions. Really started with working in a neuro-oncology lab in London when I was a master's graduate. The more I studied, I got more and more interested in how the brain works and injury and repair in the brain. And so I used to work at Johns Hopkins, where I worked with Lou Gehrig's disease. Okay, so ALS, so it's a degenerative neuro disease. Okay, and the work here for TBI. So I do all of this work in partnership with a collaborator named Tom Rao, who's also here at the University of Montana. And as I said, I worked with that protein for a very, very long time. And it started with a conversation. He looks at injury models. He looked at stroke. And we noticed that some of the things that you see in stroke were common in TBI. Okay? And so that's really how this project started. It started with a conversation in a hallway, almost about 3 years ago this month. And so that's how we started this project. And as we investigated the changes that we were interested in, the more I saw the role for this protein that I've basically spent the better part of 20 years of my life working with. And so, that's the driving force. And then, more recently, as we've gotten deeper into the field, when you meet servicemen and women, when I meet athletes that have suffered these injuries, when I meet kids that have had traumatic brain injuries and aren't recovering, aren't coming back all the way, that's the driving force to do this work. See if we can't help them.
We have another question. So you said that you were trying to decide when is a safe time to return to sports or activity. What kind of tests are you thinking that they would use in order to determine when that safe point is?
Okay, I'm going to show you the next couple of slides and you can tell me. How's that for an answer? We lost it.
Hey, sorry for the interruption, but thank you for your time, but we have to go.
Oh, okay.
So, thank you. Thank
you.
Thank you.
All right. 1 down, 2 to go. Do you guys have any questions?
Let's see. Chinook, we haven't heard from you in a while. I want to make sure that we don't, that if you have some questions, you get an opportunity. Oh, and Bozeman, I think you have to go soon too. Is that correct? Yes. Okay. Okay. So before Bozeman goes, what I'm going to say really quickly, and then Chinook, we're going to pop to you, is that if you have other questions, please feel free to email us, and I will go ahead and pass those on to Sarj. And what I can also do is, if it's okay with you, I could even send them the last few slides of your PowerPoint.
I can give you the slide deck, and you can show them.
So you guys can take a look at those a little bit more in depth tomorrow or the next day in your class. So thank you so much. I know you have to go. Now, Chinook, I'm coming to you. Go ahead.
Sorry I didn't get done in time.
Have they found a critical period for re-injury?
We're looking at that. Also, we have in our rats, like I said, we can go out 48 days and the protein hasn't recovered completely. One of the things that we're trying to do is we're doing experiments right now where we're spacing out the injuries to see if there's a drop-off point. One of the things we're continuing to do is find out, we went out to 48 days. We have experiments that are going now 6 months, 9 months. And we're going to be looking at those animals to see if the proteins came back, to get a better idea of exactly how long this is actually taking. We don't, right now, based on the experiments we've done so far, we don't know if it comes back all the way and when. Those animal experiments take a lot of time, very, very expensive. And so to keep an animal alive in an experiment for that amount of time takes a lot of paperwork. And so, we have to do it in bits that we can, depending on our funding situation and all those kinds of things. But we are trying to answer that question. All right, so this is a shout out to Caroline, because she just presented this stuff at a science fair. See ya! And I'm exceptionally proud because she got a silver ribbon for it. So that's my little shout out. So Bozeman's going to miss this part. So this goes back to one of the questions that one of the students in Bozeman just asked. It's like, is there any use for this? Is there any utility in this for determining, like, when it is safe to get back? So I'm gonna ask you, if I was to tell you that microRNAs can leave the site of the injury, how do you think we could use microRNAs how would they be useful in concussion and in TBI? Now you can't escape, Cupman, because you're on your own. Oh, Chinook, sorry. I apologize.
Sure.
Any ideas?
To remove them?
That would be a good thing. But if let's think about, before we potentially try and treat, how could they be useful in telling us about the injury? From the information I've given you so far.
They can tell you how severe the injury is.
That's definitely one of the things we can potentially look at. And going on from that, we could potentially use them to find out how long the injury is being around. Because if these things are changing and going up, is there a time when they come back down? And could we potentially use that? And one of the really important things that we can actually do with these microRNAs is potentially use them as biomarkers. Okay? So if we can detect these molecules, okay, these molecules are being upregulated in the brain, the amount of them in the brain is going up. One of the properties of these microRNAs, because they're so tiny, they can actually get into your bloodstream. Okay? So what we can actually do is potentially use these things as a measure of the injury and how long the injury is sticking around. Okay? And so that's some of the work that has come from some of the animal work that we're doing. And so we've sort of moved into doing human trials. So we're actually looking at these molecules in humans. And this is just a graph that shows a small study that we did in Grizz athletes here at the University of Montana. So we were lucky enough to be allowed to follow the Grizz football team. And when individuals, if they were suspected to have a concussion or if they were diagnosed with a concussion, we were able to take a blood sample and see if any of these microRNAs that we're interested in were able to change, or if we saw changes. And basically what this graph shows us is that if you look at people who don't play football, who've never had any kind of head injury, and you compare them to players who haven't reported an injury in 6 months to a year, that the levels of these microRNAs is incredibly low. They're almost not there at all. Okay? If you look at football players who have had concussions, okay, you can see, we see some really big changes in the amount of these microRNAs in their blood. Okay? All right? And this graph doesn't really do it justice, but we're talking like 2 to 3 to 4 to 5 times as much microRNAs in their blood compared to people who didn't have injuries. Okay? And so this is data that's for the first time showing that we can look at a biochemical marker, okay, that's a nucleic acid, that can give us an idea of the amount of injury and how it relates to the outcomes for these patients. So the difference between these two groups, and I don't want to overcomplicate things, these are football players who had concussions and still have symptoms like headaches, and then these are football players who had a concussion but then don't seem to have any long-term effects. And you can see there's a big difference between those two groups themselves. And so we think that these microRNAs might be very important in helping us potentially diagnose and then look at the long-term prognosis for TBIs. Obviously, this is a small group, and this is in athletes. We need to do. So we're conducting studies right now, all over the world, as far as Australia, with hospitals and emergency room staff to see if we can see if this holds out just in a regular population, normal population. So finally, I'm just going to finish up real quick. I know you guys, I've got you for a while. Why is this important? Obviously, knowing when it's safe for somebody to come back from a TBI, go back to their normal job, or playing sports or whatever, is really, really important. There's been a lot of this in the news this week with the NFL in terms of this long-term chronic degenerative disease called CTE, or chronic traumatic encephalopathy, that occurs primarily they've shown in athletes because that's where they've been looking. But what it seems to show is that there is some real long-term effects of multiple concussive injuries. This is a 45-year-old normal male who hasn't had any injuries or any neurodegenerative disorders, and you can see this is what a healthy brain looks like. This is from a 45-year-old male who suffered multiple concussive injuries during a career in football. Okay, you can see the difference between the two brains. It's quite striking. Obviously, this makes a very stark image, but we're trying to find out if the work that we're doing, because some of the molecules we're looking at in our rat models, we have this tissue from Boston University where they keep these brains, and we've been able to show some of the proteins and the molecules we're interested in are changing between normal brain and human brain in a similar way to what we see in our rat models. Okay, so the research that we've been doing here at the university. So one of the things we're very interested in is, like, could those potentially be the mechanisms that take a brain from this to this following multiple injuries? So protect your brain. It's pretty important.
Wonderful. Chinook, do you have any questions for Sarj?
Yeah, so for the brains that are really affected by the concussions, what happens when they're older? Do they have, like, memory loss, or?
Those are symptoms that immediately following a concussion, whoever suffered from concussion, those are things that can actually happen. Headaches, loss of memory, dizziness, all of those things. I would say in about 70% of cases, based on the numbers coming out of the CDC and some of the studies going on around the world, 70% of those people recover just fine. Okay, so the majority, as far as we can tell right now in the studies that are being done, there is a group of about 30% that seem to have long-term issues. So the headaches don't clear. They do have memory loss. They have problems concentrating. Okay, and we, unfortunately, even in the subjects that we've been working with, we've seen these kinds of effects persist and linger. And we've been doing this for about our study in humans for about a year and a half, and we're already seeing a small group that are having long-term effects. What that means in terms of our research, we're still trying to figure that out. We know we can see changes in our markers, but how does that exactly relate to why they're having memory loss? Is it because there's changes in the hippocampus? Like the images I showed you earlier. We still need to do the experiments to make that definitive link.
Good question. Any others?
I think we're all right.
All right.
Yeah, Alex, Dawn and I are still here. She's got to go, but I was wondering, this is Lennon Bozeman.
Hi, Lynn.
So, do you have some REU programs for the kids?
So what's that?
Do you know of things? Because what you're talking about is pretty exciting.
Do you have some Lynn, did you ask if there are any programs that the students can participate in? I missed that sort of.
Right. REU type programs.
REU.
Residential
Oh, residential.
Oh, okay. I know we do have at the university we have a number of programs. There's quite a few programs for undergraduates already at the university who are looking to potentially get into research. We have summer sort of internship programs. We have the summer undergraduate research fellowship program within the neuroscience department at the U. Most of the high school students that have been able to join us have obviously come from local area schools during the school year, much like Caroline in my lab. But we could look into that. I know we've had high school students come in the summer to work in labs. I haven't had one work in my lab over a summer period, primarily because my lab's still fairly new. I'm still setting it up. But I'm sure if the students are interested, we could potentially try and find some information.